We're almost there. Just have to. Yeah, everything's sluggish. Are you guys having trouble connecting to Blackboard today?
Yeah.
Yeah, some people are saying that. Starting now, I'm going to share screen. All right, fine. All right. Okay, it's told me that you guys get up to about here. Let's see if you guys agree. Was it around here that you guys found it last time? So functions of membrane proteins. So within the lipid bilayer, just as a quick review of what we were talking about, so we get back into this. So you have the lipid bilayer, and it's often referred to as a fluid mosaic model. Was that terminology used? So a mosaic is like, if you think of like ancient Rome, they used to take little colored tiles and stick them in a kind of cement, and then make a beautiful picture of an animal like a lion or something. So that's the idea of a mosaic here. So the lipids are like the cement, and they're actually pretty mobile. They can move around laterally. Occasionally, a lipid will actually flip from one side to the other. And then the protein would be the equivalent of the tiles. They're in there, and they have a lot of different functions, some of which are shown here. But they can actually float around. They can actually move laterally within this sort of sea of lipids. Now, if need be, a cell can actually tie up protein down in one location by having, Dr. Bach probably talked about the cytoskeletal elements, right, within the cell. So you have actin filaments contacting these proteins and holding them in place. You may actually have proteins also contacting extracellular elements, which I think Dr. Bach talked about last time also, right? Holding them in place, okay? So here's just a quick review of some of the things we talked about. You probably talked about channel proteins, which are pores. What is that? Well, it's kind of shown here, but what would you use your channel proteins for? What's the purpose of them? What kind of molecules or things, atoms, can go through a channel?
Water molecules?
Yeah, you have aquaporin channels, right? So those go through channels. The is water polar or non-polar? Polar, right? So water has a difficult time getting through the hydrophobic core of a lipid bilayer, so that's why we have channels. What's something else that has a difficult time getting through the lipid bilayer without the aid of a protein? Sodium, right? And when we say sodium, we're talking about the sodium ion. Same thing with potassium, it's in ionic form, as well as chloride, things like that, calcium also. So we have specific channels for all those things, which are essentially pores made out of protein. We also have carrier proteins. Well, unfortunately, they give me too much information, but it tells you right there. We have glucose, right? Glucose is a 6-carbon organic molecule. It's polar. Why is glucose polar? Anyone remember? What about its chemical nature makes it polar? Sugars have a lot of what kind of group on the outside of them? Well, on them.
Hydrogen.
Yeah, well, hydrogen, but also hydrogen combined with oxygen is a hydroxyl group. Remember that? So oxygen is our oxygen more electronegative or less electronegative than hydrogen?
More.
More, right? So if it's like a molecular tug of war, oxygen will pull on the electrons more than hydrogen. Same thing with carbon. And so you have a partial negative charge on the oxygen and a partial positive charge on hydrogen and carbon. So something like glucose has a very hard time getting across the membrane. That's also kind of relatively big, 6-carbon backbone. So it needs a carrier. So we have the gluc carrier. And then we have something like this, the MHC, major histocompatibility complex glycoproteins that sit in your membrane. So all the cells in your body have these glycoproteins. And they're involved in things like cell-to-cell recognition. Also, your immune system recognizes your cells and doesn't attack them, but will attack bacterial cells or viruses, right, based on the surface proteins and surface carbohydrates found there. Now, this term glycoprotein, when you look at the word glycoprotein, obviously you know what a protein is now, right? What does the glyco refer to? What's that? Related, it's related. So the glycogen, the glyco in the word glycogen and the glyco here in glycoprotein mean carbohydrate, right? So glycogen, you're right, it's a carbohydrate, right? So it has the same, so it's a prefix that whenever you see glyco, you want to think carbohydrate. So it's a carbohydrate attached to a protein. All right, so let's pick it up. Okay, so receptor proteins have a shape that allows for specific molecule to bind. So here's a receptor protein. So all the cells in your body have these receptor proteins, and they'll have a very specific little cleft here that has a specific shape. And it's going to typically just going to bind to a very specific molecule, which could be a neurotransmitter, it could be a hormone, something like that, that will bind to it. And then what happens is once this molecule binds to this protein, this receptor protein, a shape change will occur in the protein. And there's actually a huge variety of different things that can happen. Basically, the protein will change shape, and this can trigger a number of things that can happen within the cell. Okay. There are also enzymatic proteins. Here's an example of this. On the cytoplasmic side, facing in towards the fluid on the inside of the cell, these actually have a very special little location here, and you can actually help. It'll catalyze reactions, so it acts as an enzyme here, right? So you could take a molecule as is shown here, the molecule will bind here loosely through things like hydrogen bonds, and then it will be actually converted into another type of molecule. So this is a protein in the membrane that acts as an enzyme. An example here would be the proteins electron transport chain. These are found in mitochondria, and they're involved in aerobic respiration. All right, let's go on. Here's a question. So, which is a correct association of protein types? So go ahead and talk to your neighbor if you want to. Think about what the correct answer is here, and then we'll give you a moment to think. Anyone want to volunteer? No? You're still thinking? Okay, I'll let you think. Okay, let's look at them in order then. Cell recognition proteins, right? Allow a specific molecule to bind to it and they cause a cell response. Let's go back to here. Allowing a specific molecule to bind you, that's a characteristic of a receptor protein that leads to some sort of response within the cell. So that one's actually not this false. Okay, let's go to the next one. Protein channels combined with a substance to help move it across the membrane. Is that true or false? What do you guys think? True? No, sorry. So thank you for volunteering, but it's actually, this is why we're doing this, right? So there's a lot, people get confused between channels and these carrier proteins, okay? So in a channel, the molecule, be it water or sodium ion or potassium ion, never binds to the channel. There's definitely interactions between the ion and the walls of the channel, but it's never really binding. They actually can move through at a very high rate, okay? So when you have, for example, aquaporin channels, I mean, the water molecules just pile through very quickly. They're never really actually binding, right? Whereas with the carrier protein, something like glucose will sort of, it has a certain affinity for a site in this carrier protein, and it will sort of temporarily bind. In a sense, again, for very weak bonds like hydrogen bonds, things like that. And then this then, once it binds, it induces a shape change in the molecule. Because the molecule then the carrier molecule changes shape, and then the molecule trying to get across the membrane actually moves straight across. So, for example, you can look at it this way: you have, and this is just a schematic. It doesn't actually look exactly like this, but you have your carrier protein sitting in the membrane. The way I always draw this is the inside of the cell downwards and outside of the cell top here. Now imagine you have glucose. It comes along. We've got a bunch of glucose out here. and one happens to bump into here, right? So it sort of temporarily binds. And then in the next instant, what happens is you have a shape change, which I'll draw over here. So we can imagine this molecule then goes through a shape change. And now the glucose, which was outside of the cell, ends up inside. And then, these things, again, they're not bound, like forever, right? So they're just loosely bound. And so now it can be released to the inside of the cell. Okay. So with these carrier proteins, there's never a point in which there's an open channel between the outside and the inside. Okay. Whereas with a channel, there can be an open channel going right through the middle. All right, let's get back to it. Whoops, I just needed to answer, didn't I? All right. Receptor proteins, variations of these proteins cause tissue transplant rejection. What kind of protein would that be? Let's go back to our picture. That'll be something like this, a glycoprotein, right? Because those are involved in cell-to-cell recognition. So neighboring cells can recognize each other, and the immune system can recognize your cells and differentiate between your cells and something else, like a protein coat of a virus, or the protein or carbohydrates on a bacterium. So that leaves this one. So which is the correct association? This has to be the right one. Enzymatic proteins carry out metabolic reactions directly. Okay, so they have this bit that's hanging down here into the cytoplasm, and they'll carry out a metabolic reaction. Okay, so let's move on to membrane permeability. So we're talking a lot. This whole chapter is essentially on the plasma membrane because it's an incredibly important part of your cells. It's incredibly important for life. One characteristic of the plasma membrane is it's selectively permeable. This means some molecules can freely cross the membrane, but others can't. Okay, so and the ability of something to get across your plasma membrane depends on its size. So smaller molecules can get across easier than large molecules. It also depends on the nature of the molecule. So for example, I already mentioned this earlier, and I think we have a little bit of background on this. Something that's polar, like water, can't get through the plasma membrane very well unless there's a channel. Something that's ionic, like sodium, which has a charge of +1, it can't get through the membrane without having a channel of some sort. However, on the other hand, small uncharged molecules can freely cross the membrane. So oxygen can get across your membranes very easily, right? So all of your cells need some, well, vast majority of your cells use aerobic respiration, so that means that you're using up oxygen, right? So luckily for us, oxygen can very freely diffuse right across the membrane, okay? Now, why is that? Well, oxygen is an electronegative atom, but here we've got oxygen paired with oxygen, right? So they're having this molecular tug of war with each other, right? And they're kind of equal. So this means that the oxygen O2 is a non-polar molecule. It's just, it has a double covalent bond. It just looks like that, right? So it's linear and it's non-polar. What about CO2? Does anyone know what CO2 looks like? What's the molecular formula of that? It's carbon and 2 oxygens, right? Mm-hmm. So you may have seen this, at least in passing in another class. It actually looks like that, right? In chemistry or something. Carbon's in the middle. Now, oxygen is more electronegative than carbon. So you might think, well, hey, this should be a polar molecule, right? That's not a bad thought, but it's actually non-polar. And the reason for this is because it's in a straight line. So it has because of that, it's not really a polar molecule. So now there's some other ones that are actually technically a little bit polar. Glycerol and alcohol all have hydroxyl groups, OH groups hanging off them, but they're small enough that they can actually get through the membrane. They have a little bit of a hard time, but they can get through the membrane. So again, it's a little bit of a combination of these different factors that determine whether something can get across the membrane. Okay, so these smaller things can slip between the hydrophilic heads and pass through the hydrophobic tails and get into your cells. And they're driven by the concentration gradient. So they're going to move from an area of high concentration to an area of low concentration. Let's use the example of oxygen. Your cells are constantly using up oxygen, right? Where does oxygen get used up, and what organelle is the oxygen used up within your cells? They're referred to as the cellular powerhouse, you can say.
Respiration.
Yeah, through aerobic respiration but what organelle then? Yeah, anyway, mitochondria, yeah, you're right, you're on track. So they're going into the mitochondria, right? And oxygen's disappearing, and oxygen actually combines with hydrogen and becomes water, H2O, right? So oxygen's entering your cells, it disappears all the time. So basically, for most of your cells, you're going to have more oxygen outside than inside, right? So there's a greater concentration of oxygen outside compared to inside. So oxygen, just by will move down its concentration gradient from an area of high oxygen concentration outside to an area of low concentration inside. Actually, so okay, well, I guess we've got a little ahead of ourselves before, right? So water is a polar molecule. It cannot readily cross the membrane. We talked about that earlier, right? So the way they get across is through these aquaporin channels, right? Sorry about that. I got mixed up. I thought you got the aquaporin channels right, but you understood what I was talking about earlier, right? Okay. So we're getting it to here now. Large molecules, ions, and charged molecules are unable to cross the membrane freely, but can cross the channel proteins. So we actually, I got a little bit, jumped the gun a little bit. Those are usually these pores to the membrane, we just talked about. And then there are carrier proteins, and I drew the glucose transporter or carrier protein on the board over there. That's going to be very specific for the substance that they transport. So a glucose transporter will carry glucose across the membrane, but it will not, for example, move sucrose across, right? Sucrose is bigger, it's a disaccharide, whereas glucose is a monosaccharide. And then we're going to talk about vesicle formation. There's also very large molecules like proteins, and some polypeptides are relatively large. They're going to have to move across the membrane through two cellular processes. One's called endocytosis, the other is exocytosis. Okay, so here's a bit of a summary of some of the things we were talking about earlier. So this is a good thing to study and understand this diagram. So we have charged molecules. You see how this arrow is bending around. They're indicating that the charged molecules cannot go through this nonpolar hydrophobic core because the charged molecules actually like to associate with water. Are you guys familiar with that idea? Why would a charged particle like sodium be so attracted to water? Did Dr. Bach draw this on the board for you? So you have sodium. Let's imagine here's sodium. And it's inside the cell. Inside the cell here. Now it's surrounded by water. So tell me which is the correct orientation for water. Here's one water molecule. Here's another one. Now, I understand the water molecules are bouncing around, there's thermal energy, so nothing's really static. But if you were to just somehow have a magical camera to take a picture, which would be more likely? Having oxygen this orientation with the hydrogen end is towards the sodium ion, or the oxygen with the oxygen water molecule with the oxygen pointing towards the sodium. What do you guys think?
Second one.
Yeah, this one too, and you can probably get around. Yeah. So remember, because this has this little Greek letter delta, it has a negative, partial negative charge. You have the Greek letter delta here, these are partial positive charges. So, I mean, this definitely happens occasionally because these are moving around all the time, but it's probably more likely that this negative end would be pointed towards this. So sodium. So what we often think about this sodium is not really by itself, but it has what's called a hydration shell. Whoops, there should be a negative here, which means it has sort of its accompanying water molecules. Okay, so it really likes water, and it does not really like things that are non-polar very much. So that's why it has such a hard time getting across the membrane. Water molecule, for similar reasons, water is a polar molecule. It's attracted and it likes to make hydrogen bonds, which you've heard about before, right, with other water molecules, but it doesn't really interact so much with non-polar molecules. Now, what about something like a fatty acid? You learned about fatty acids before, right? What's a fatty acid look like? in terms of molecular structure, what's it look like? It's very long, right? What's that long tail made out of? Go ahead.
Phospholipids.
Well, it's no, but what atoms is what I'm trying to. In a fatty acid, what's that long tail of a fatty acid? It consists of two atoms.
Carbon, hydrogen.
Carbon and hydrogen. So hydrocarbon tail, right? You heard that terminology right before. And at one end is a carboxyl group, which is COH. That is in fact polar, right? But the most of a fatty acid is in fact nonpolar, and it's small enough they can sort of slip through the membrane. Okay, so there's some molecules like that can slip through. Here's a macromolecule. It's a protein. Proteins are large. They also often have a negative or positive charges, right? Depending on the side. Remember, you learned about the side chains of the R groups of the amino acids. So those can have positive charges, they can have negative charges, they can be polar. All that makes it difficult for the protein to get across. Plus, they're pretty gigantic molecules too. Okay, so all right, so we're gonna now. So that gives you a little bit. We've gone through how things get across the membrane. Let's talk a little bit about this process of diffusion, which is really important for our cells. So I mentioned earlier that things will move down their concentration gradient, like oxygen will move down its concentration gradient and move into your cells. What's the actual mechanism by which this works? Well, and that's kind of shown here. What they've done is they put some purple food coloring into a sugar cube, and they placed it in the bottom of a fishbowl. I don't know why they've got it in a fishbowl. It's mean to the fish. Just probably the artist was a little bored, okay? But what we have here is a very high concentration of dye molecules, right? Over here, and other places we have essentially just pure water, or close to pure water, so a high concentration of water molecules, H2O. Over time, you would all expect this to happen, right? This would dissolve, and the dye would eventually get, after about a day, or maybe even half a day, the dye is sort of spread out completely, and they're intermixed with the water molecules, right? And it's very unlikely for everything to go back the other direction, right? So this is just a sort of random process due to thermal energy within the water. Okay, so basically what we're seeing here is the dye molecules are moving from a region of high dye molecule concentration to an area of low dye molecule concentration. And eventually the process stops when the dye molecules are even spread out, so there's no difference in concentration. It can't occur anymore. But what might be not as clear is here we have water molecules here. There's a high concentration of water molecules here, and a low concentration of water molecules here inside the sugar cube, right, with the dye molecule. And so water also wants to move, in quotes, because obviously the molecule can't want something, but it's going to move down its concentration gradient into an area of low water concentration. So the water molecule also is moving this way, right, at the same time. Okay, so that's the basic idea of diffusion. And we use the term net diffusion to indicate when something is moving down its concentration gradient, the change in concentration of a molecule over time. So why is this important? Well, if we look in our lungs, we have these tiny sacs called alveoli. And the individual one's called an alveolus. We have oxygen within our lungs, and it has to get into the capillaries, the pulmonary capillaries. These are the lung capillaries. So how does this work? Well, oxygen is in a higher concentration within the lungs, the airspace of the lungs, and a lower concentration within the capillaries. So there's a natural tendency of oxygen to then diffuse down its concentration gradient into the blood plasma within the capillaries. Now CO2 is in a higher concentration within your blood plasma, which is the fluid around the red blood cells within your capillaries. And so CO2 will go in the other direction, okay? Now, there's a number of factors that'll affect the rate of this diffusion. One is temperature. The higher the temperature, the faster the rate of diffusion. The higher the pressure, the higher the rate of diffusion. Electrical currents can affect diffusion if we're talking about the movement of charged particles, such as sodium, which is a charge of +1. It's not going to have any effect on oxygen because it's not charged. And molecular size, I already mentioned that earlier, but small molecules will diffuse easier compared to large molecules. Okay. All right, let's take a look at this video here.
Diffusion is the net movement of molecules down a concentration gradient. This process allows small molecules such as oxygen and carbon dioxide to cross the plasma membrane. Most polar molecules, such as sugars and proteins, cannot freely cross this lipid membrane. Although water molecules are polar, they are small enough to pass through the membrane freely. This special case of diffusion that involves the movement of water molecules across a semi-permeable membrane is called osmosis.
Let me stop that for a second. So the idea here is we have a plastic membrane and it's porous. The pores, as you said, are just big enough so that the water molecules can get through, but they're quite small pores. So this next molecule won't be able to get through.
If a molecule such as urea is added to one side of a membrane, it will not be able to diffuse across the membrane because it is both large and polar. Because of its polar nature, it will interact with other polar molecules such as the water. This interaction reduces the number of free water molecules on the right-hand side. Because the concentration of water molecules is lower on the right-hand side, there is a net movement of water molecules down their concentration gradient to the side with the urea molecules. Because more water molecules are moving into this area than are leaving, the water level on the right side will rise. If the osmotic concentrations of two solutions are equal, the solutions are isotonic. However, when the solutions have unequal osmotic concentrations, the solution with the higher concentration of solutes is hypertonic, and the solution with the lower concentration of solutes is hypotonic.
Okay, so there's kind of an interesting thing. So let me just stop it here, this video here. So the water, there's people see that the water concentration was initially higher on one side than the other, right? And water moved down its concentration gradient just by diffusion right across the membrane. And whoops, it's touch sensitive. So then the water moved up. So will this continue to happen? Because here we have solutes here, right? Will it just continue to keep moving forever, or what's going to happen here? Or does it actually stop? Did anyone see this in high school, a demo of this or anything? Yeah. So does it keep going forever, or does it stop?
It stops.
It actually stops, right? Well, why does it stop? Does anyone know why? Well, what happens is you actually have pressure. We actually refer to the term osmotic pressure, and it's a true pressure. Pressure is actually force per surface area. So the water, actually, you have a water column that's higher here than over here. That has a certain weight, it's pushing downwards, right? And it actually ends up pushing the water molecules back, pressure ends up pushing the water molecules back at a certain rate. Whereas the water molecules are moving down their concentration here at a certain rate, eventually get to the point where they're equal out, and this doesn't go any higher. So that brings us to this next slide here. So this is exactly how you would actually do the experiment. And we have a special term, osmosis is essentially just diffusion of water, right? But the added trick to it is that it's always across some sort of selectively permeable membrane, right? And that could be a membrane such as your cell membrane, or it could be this plastic membrane we're talking about here. So we have diffusion of water across a differentially permeable membrane. It occurs from an area of higher to lower concentration. It occurs from an area of higher water concentration to an area of lower water concentration. Now, what that means effectively is the water is moving towards an area with higher solute concentration, right? And we saw that. We saw that in this video, right? It was moving. The water was actually moving towards the area with higher solute concentration. Since the solute can't cross the membrane, it draws the water towards it. So in a sense, the water is moving down its concentration gradient. It appears as if the solute is drawing the water towards it. It's not really. The water molecules actually don't know anything. They're not actively drawing something, right? But this is what it appears to be doing. Because the water concentration is lower in an area with high solute concentration, the water will move that way. And you can actually see this here. We've set this up. We have this flask at the bottom. There's this porous membrane. Here we have lower water concentration because we have a solute in here, and a higher water concentration out here in the beaker. So water will move down its concentration gradient. So it's effectively moving towards the area of higher solute concentration, which is what we're talking about here. So osmotic pressure is the pressure that develops due to osmosis. So you can see that this water has moved up. And we could weigh this amount of water. We could say, well, how much pressure is being pushed downwards on here, and that'll actually stop at the point where that pressure is in fact the osmotic pressure. Okay, so it's something you can measure quite easily in these sorts of experiments, because the pressure will actually physically push the water molecules back across the membrane. Okay, so that brings us now to tonicity, which you might have heard about previously. It refers to the relative concentration of solute inside and outside of the cell. So a quick caveat, a warning I have to give you is this, what I'm going to talk about here works perfectly as long as we're talking about solutes that are non-penetrating, because that's actually the technical term. The members we're talking about here, be it biological or a plastic membrane, for these solutes, they're non-penetrating. The solutes cannot get through the membrane. So the only thing that's going to be moving here is going to be the water. It can get through the membrane of a cell in your body because we have aquaporin channels, or if we're talking about a plastic membrane, it can go through the plastic membrane, through the pores. Okay, so in this particular example, we're going to use a red blood cell. So let's look at isotonic solution first. We have a solute concentrate, a situation in which the solute concentration is equal inside and outside of the cell. Let's draw a picture of what this looks like. So imagine like you're a doctor or a scientist, you have a beaker filled with water, and you have a microscope, you put a red blood cell in here. So red blood cells, if you look at the side of them, they're biconcave, right? You might have read about that. So in this case, if you look inside and outside of the cell per unit volume, imagine like a little square of fluid. I'm trying to make them roughly equal, right? These are the solutes, the little dots, right? There's the same concentration of solutes inside and outside of the cell. Now tell me about the water concentration. Is the water concentration higher outside of the cell or inside of the cell? Or is the water concentration equal? What do you guys think?
Equal.
Yeah, it's equal, right? So that means you're going to have some water entering through aquaporin channels, right? But you also have some water exiting through aquaporin channels, right? There's thousands of these channels. Now, would you expect your cell volume to change in this situation?
No.
No, so it's going to stay the same. So our cell, it neither gains nor loses water. I mean, water's moving across, but there's no net gain or loss. Okay, let's look at the next one. A hypotonic solution. A solution has a lower solute. The solution, in this case, the solution has a lower solute concentration than inside the cell. Okay, hypotonic. So let's take the same diagram and just adjust it a little bit. So I'm going to put I'll just make it pure water to make it so we don't have any solutes, right? That would be easier. So our solution is hypotonic relative to the cell. Now, what do we expect to happen here? Well, let's look at water concentration. Where is the water concentration higher, inside or outside of the cell?
It'll be higher inside.
The water concentration is higher inside?
If it's hypotonic, doesn't it make the cell go bigger because the water is being drawn inside the cell?
Right, bro. That's correct, but you're one step ahead. I want you to do logically, step by step. Where's the if you took a little volume, when I say volume, like imagine a little square like that. This is pure water, right? Because I took all the solutes away, right? Now, if I draw a little square in here, there's less water because there's some solutes, right? So the solutes, they physically take up space. As the video you saw, they actually attract the water and reduce the free water concentration because they're sort of binding loosely with hydrogen bonds to the water. So in this case, there's a higher water concentration outside than inside. So what do we expect water to do? Will they be a net water movement of water into or outside of the cell? You actually said it just a second ago, so just repeat what you said.
It goes into the cell.
Yeah. because the water is gonna go into the cell, right? There will be some water moving out, right? Because water is just randomly gonna go through aquaporin channels out. But overall, this is supposed to be a big arrow. There's gonna be water moving in. So over time, what's our cell gonna do? Is it gonna increase in volume or decrease in volume?
Increase.
It's gonna increase, so it'll puff up. And eventually it might even lyse, which means it just breaks apart, right? Lyse is to break apart. All right, so that's hypotonic. The cell gains water and swells out, lysis of the cell may occur. Now let's look at hypertonic. Okay. So here's the last one. We have our red blood cell. It's awfully big red blood cell, right? Relative to the beaker side. Hypertonic. Now that means a red blood cell has to have solutes. Now, I should have mentioned this earlier, but when we talk about solutes, we're talking about essentially particles, right? So what we're really counting is the number of particles. So it could be sodium, potassium, chloride, glucose, proteins. Each one of those counts as a single particle. That's what we mean by solutes. So the cell has to have some of that because otherwise it'd be dead. Now, I'm not gonna, it would take me a whole hour to fill in, but imagine, so I'm just doing it right around here. We're hypertonic, meaning it's just absolutely chock full of, say, sucrose or something like that. Maybe that's our molecule, right? So there's a very high concentration of sucrose, or this, which is our solute outside the cell compared to inside. So is the concentration of water higher inside or outside of the cell now? What do you guys think?
Inside.
Yeah, so if you look at a little volume here and here, clearly there's going to be more water inside because there are fewer solids, right? So water wants to move, again in quotes, right? But it wants to move down its concentration gradient. So we're going to get a net movement of water in which direction? Outside or inside?
Outside.
Yeah. So water is going to move this way, right? And of course, I put the water right here because some water, of course, will go the opposite direction, right? Just by random chance through these aquaporin channels. But overall, we're going to get water moving out. So our cell will shrink, right? Okay. So it's going to shrink and you get this process of crenulation where the red blood cell will get really spiky looking. Now, plasmolysis occurs in plant cells. Have you heard of the term plasmolysis before? So remember, in a plant cell, you have a cell wall that's made out of cellulose. And then within that, you have a plasma membrane. And they look kind of like this. Now, if you shrink your plant cell, you won't shrink the cellulose won't shrink, right? But the cell will. So the cell will essentially, it only has so much membrane. So you then get these gaps. That's plasmolysis, right? Okay, so that's what's being shown here. So we've basically gone through all the different steps here, right? We looked at isotonic, hypotonic, the cell will increase in volume. Hypertonic, the cell becomes crenulated and decreases in volume. Similar things happen with the plant cell. One of the biggest differences here is that because the plant cell has this hard cellulose surrounding it, the cell wall, then the cell membrane will essentially break away from that cell wall. Okay, let's do a question here. If you go to any hospital, you see this type of a solution. Okay, it'll be called it's a fairly standard isotonic solution. It's 0.9% NaCl. Now, first of all, the people know what that means. When we do a percentage, this means that we have it's by essentially by weight. So we're saying we have a little bit less than 1% by weight NaCl. So it's the weight of the solute compared to the weight of the solution. Okay, that's what that percentage sign means. Okay, so this is isotonic with that amount of sodium chloride. That's just the normal salt you find in any kitchen, right? That's what we have for cooking. If 0.9% NaCl were isotonic to a cell, then what do you guys think? is the correct answer here.
Is it the fourth one?
Yeah, okay, let's look at the fourth one. 1% would be hypotonic to the cell.
Wait, no, it's the third one.
Third one, yeah, that's better.
I read it wrong.
It would be hypertonic, right? So it has more solutes than the cell. So if it's hypertonic, then our cell would shrink at least a little bit, right? Okay. So the vast majority of when you go to a hospital and they give you an IV, typically that's going to be isotonic. There are hypotonic and hypertonic possibilities for very specific clinical reasons, right? Those do exist, okay? But the vast majority would be something like this solution, the 0.9% NaCl is isotonic. So 1.0% would be slightly hypertonic and cause your cells to shrink just a little bit. Okay, let's talk about transport by carrier proteins. So there are different types of, so we talked about earlier why some molecules can't get across the membrane, right? So in order to help them across, we need to facilitate their transport. So the term facilitate just means to make easier, okay? So facilitated transport explains the passage of charged molecules. Remember we talked about sodium, which is charged +1, for example. Glucose, it's polar and relatively large, it's 6-carbon organic molecule. Amino acids, right? Those are fairly large also. And they can be charged or polar. So they can't get across the membrane very easily, even though there might be a concentration gradient individually for each of those types of molecules. So what we have then is different ways of getting things across. One is one form of facilitated transport is the ion channel, which we talked about earlier. The ion channel makes it easier for ions to get across. Also, the aquaporin channel for water. You have very highly specific carrier proteins that undergo shape changes, right? We talked about that already, actually. We talked about the glucose transport. It's a typical one that people talk about, but it's so important. It helps to bring glucose into our cells. Now, these don't require any ATP because they're basically the movement is based on the fact that there's a concentration gradient. As I mentioned earlier, towards the beginning of this class, I said typically you have more glucose outside of your cells than inside because your cells are using up oxygen. Using up the glucose all the time because they're converting it, they're using it up to make ATP, right? You learned about that earlier. All right, so this anything that like this is facilitated transport is a form of passive transport. It doesn't require energy, right? So when you heard passive, it doesn't require energy. It's the equivalent of having like a ball at the top of a hill and just let it roll down, okay? So these molecules are moving down their concentration gradient. You do not have to supply any additional energy other than just the regular thermal energy that's already in there with the molecules bouncing around within your cells. Now there's another form of transport called active transport. This is where this can also transport things like charged molecules like sodium against its concentration gradient. And it can accumulate something like, for example, you can accumulate potassium inside of cells, you can accumulate sodium outside of cells. Active transport is highly specific. It requires these special carrier proteins and energy is required, often in the form of using up adenosine triphosphate and hydrolyzing it to ADP, adenosine diphosphate, and an organic phosphate. So here's an example of facilitated transport. We have I don't like the way the artist did this. They made it look like a channel, right? But it doesn't really look like that. Here's a solute that goes in here and it moves across the membrane. I actually drew a picture of this earlier today. Then there's active transport. Here's a really classic example of active transport that you all need to know. Have you heard of the sodium potassium pump before in a previous class? Some people are saying yes, some people no. Okay, so this is one of the most important pumps in your whole body. So we do have to understand that it's going to come up over and over again. So what I'm going to draw here is a picture of a cell in your body. And I'm not going to draw all the details they have here. I'm just going to draw the pump as it's essentially a protein sitting in your plasma membrane. And it's going to pump potassium into your cells, okay? And at the same time, this pump, for each cycle of the pump, it actually pumps sodium outside of your cells, okay? So there's a mnemonic to help you with this. You just have to remember the word pumpkin. I think, do you see where I'm going with this? Pump, right? Pump. K in, right? So that's what the sodium potassium pump, also called the sodium potassium ATPase, does. Now this pump requires energy. So it's going to take ATP, it's going to hydrolyze it, and you'll have ADP and plus inorganic phosphate, which is PO4 minus, right? So as we go through this in many cycles, what's going to happen? Well, we're going to have a lot of potassium inside of your cells. So I'm just drawing a really large K to indicate a large potassium concentration inside of your cells and a lower potassium concentration outside of your cells. We could use the square brackets that indicates concentration, so you can put those on here too. Now, if we're pumping sodium outside, what's going to happen here? We're going to, over time, we'll have a very large sodium concentration outside and a smaller sodium concentration inside. Okay, so this is a major pump in your body. It's working tirelessly all the time. So if you look at your brain, it's very metabolically active. About 70% of the energy your brain uses is just for this one pump. Okay, so we can look at the details here of how this works. Step one, the carrier. Oh, are we out of time? No, we have a couple minutes here. Okay. The carrier has a shape that allows it to take up 3 sodium. So there's little spots for the sodium to bind here, right? Next step 2, ATP comes along, it's split, and a phosphate group attaches to the carrier. You then have a shape change, you see this? And the 3 sodiums, they're only loosely bound here with things like hydrogen bonds, right? They can come out. They release the sodium to the outside of the cell. The convention here is the outside is always on the top, right? Here's the inside of the cell. The carrier has a shape that allows it to pick up potassium. So it's picking up 2 potassium. And then once that happens, once they bind, it changes shape again. The phosphate group is released, and now it changes shape, and then the potassium ends up inside. Okay, so it cycles around over and over. It's tirelessly doing this, all day and night for you all the time, and it's maintaining this difference in gradients across the membrane. Okay. All right. So one thing you should note here. Remember this difference in numbers here, right? So it's 3 sodium for 2 potassium. That's something you should probably also know. One way, the way it helps me remember this is the fact that the inside of your cells are actually slightly negative compared to the outside. And so how could that be? Well, if you're moving 3 sodium for every 2 potassium, what are we doing here? We're moving for each cycle of this, right? We end up moving 1 positive charge outside, right? Compared to the number, the difference, right? Between 3 minus 2, right? So that's going to make the inside of the cell slightly negative. Okay. So this channel is what's referred, this crop is referred to as electrogenic. It actually helps to contribute to the inside of the cell being slightly negative compared to outside. All right. So yeah, I'm not going to be able. So let me just quickly mention this, and I think Dr. Bach will have to finish this up next time. So far we've talked about relatively small molecules like glucose, water, ions, things like that. What about much bigger things? How do you get a protein across the membrane? How do you get a peptide that has remember peptides are chains of amino acids? What if it contains 10 amino acids? Well, at that point, you need to do something different. You're going to use bulk transport. So we're going to move large macromolecules, such as food particles, hormones, waste that are too big to be transported by carrier proteins. They have to move by bulk transport. So they're transported into or out of the cell by actually vesicles. Vesicles are little spheres of membrane. It's going to require a lot of energy to do this. So this is a form of active transport. And there are going to be 2 types of vesicles that are formed that do this membrane-assisted transport. There's exocytosis. Exo sounds a lot like exit, right? So it's going to help these substances exit from the cell. And we're going to have endocytosis. Endo means in, so it's going to bring things into the cell. So endo means in, cyto means cell, well, site means cell, and osis is a process. So it's the process of bringing things into the cell, and the opposite for exocytosis. Okay, so that's a little preview, and Dr. Block will have to finish that up next time, right? So he's gonna go into forms of endoexocytosis. And then we're very close to the end of this chapter. Okay. Thanks, guys. We'll be back to your normal teacher next time, right? All right.
I got told that I was in today.
Okay, I'll take it for you. Thank you. I'll put it up. Thanks.
Thank you. Have a good day.
You too.
Hey Jerry, when's the meeting down? Yeah. Are they gonna be ready for us? Yeah, I told Abby yesterday.